Hello, welcome to the Omics cast. And in this episode, I will be talking about ONT targeted epigenomics. My name is Alberto Vilella, and if you want to follow me on Twitter, my Twitter handle is Alberto Vilella, all one word. So you can follow me on twitter.com/Alberto Vilella, all one word. And you can also contact me on LinkedIn. Here's the link to do so. And I welcome people contacting me, asking me questions of any kind. This slide deck is available upon request. In this episode, I'm going to be talking about the analysis of a paper on ONT sequencing of targeted regions and the epigenomic analysis or epigenetic analysis that they do of the methylation marks on those targeted regions, and this happens in the human genome. The authors, Winston Timb, and as you can see here, there's also people from Nanopore, Oxford Nanopore, the company themselves, contributing to this paper. Winston Timb and Jared Simpson have been doing a lot of work on the calling of epigenetic marks from Nanopore reads, and you'll see an example of this in this paper. This was published in bioRxiv very recently. So what they started with is about 3,000 nanograms of genomic material. And they do this in this paper in cell lines, which can grow on a Petri dish, and you can produce a certain amount of them so that you can extract here 3,000 nanograms of genomic DNA for each of them. And then they achieved an enrichment on the regions where they did the targeting of between 10 and 300 fold, right? Meaning what they get genome-wide for what they sequence. In the regions that they targeted, they get 10 times or even 300 times more than that. And all regions were evaluated from a single flow cell, and the coverage was ranging between 20x and 800x. They did this on mean ion flow cells, although they did it on a grid ion. But to achieve 800x coverage over the human genome, you would have to do several promethion flow cells, whereas here with a single mean ion flow cell, they get coverage ranging from 20 to 800x at the regions of interest, the sites of interest. Total yield per flow cell range between 70,000 to 230,000 reads, and the on-target fraction, meaning all the reads that align to the region compared to the total fraction of reads, was between 1.7 and 7.6%, which is, 7.6 is a nice number. People that have done targeted enrichment with other technologies, they know you can get it to 10%, 50%, 15%, maybe 20% or more. So achieving 7.6% is quite a feat here for a technology that it's still evolving. The off-target reads seem to be distributed randomly across the genome, indicating that they result from ligation of nanopore adapters to random breakage points, right? And so they show how they do this, and then they hypothesize where the off-target comes from. This is an illustration of how they did it. They have a region of interest, ROI, region of interest marked here in green, and then they design Cas9 targets to cut the upstream and downstream sites flanking this region. And then they do this on dephosphorylated ends, so that the phosphorylated ends that remain are only those from the cut. And then the library prep, as you can see here, in theory, should only put adapters to phosphorylated ends coming from the cuts, right? Because all the other ends that are not from the Cas9 cut should have been dephosphorylated. And what they say is that this phosphorylation is probably not perfect, and it still leaves some phosphorylated ends, or ends that are not phosphorylated but yet receive the ligation that would create off-target reads. But what they want is to maximize the amount of this type of inserts that receive the adapter and then can be sequenced. And so here's an illustration of reads coming from both ends and in different colors. And here is how they show the coverage going up or down across the different cell lines here, 4 different cell lines for a given region of 18 kV. And then we move to methylation. They say that 5 of our selected loci are in the promoters of genes where expression level and promoter methylation have a prognostic implication in human cancer, right? And so what they're trying to say is, given that the Nanopore sequencer gives you the raw signal, the squiggle signal, that will look different if it goes through a methylated C, a C of a methylated CpG, then you will be able to identify that. And in the same way that you're sequencing all the read in that insert, you'll also have the signal for bases that are not ATCG without methylation or other marks. You'll also get the methylated reads. And so they compare their results with results they previously had generated with the, if you want, old school technology of whole genome bisulfite sequencing, WGBS, for whole genome bisulfite sequencing. And they do this in 12878. And they say that they see very similar methylation patterns between the targeted Nanopore and WGBS data analyzed across the regions where they did whole genome in bisulfite and targeted in Nanopore. Pearson correlation of 0.84, which is a decent correlation, considering that some of these can be just low coverage on a given region for the whole genome bisulfite data. And they observed that in their own Nanopore reads, because these reads are long, they can be several kilobases long, they see clusters of completely methylated or completely unmethylated, if you want, haplotypes, right? So examples of useful biological information from this is what they see a dramatic increase in methylation at the promoter of this gene in a given cell line, or the change in methylation in this other gene. One remains largely methylated in non-tumorigenic cell lines, but the gene promoter becomes hypomethylated in both of the transformed cell lines. So looking at RNA-seq data that they produce for this, or that is publicly available, they demonstrate that the mRNA levels of these genes follow the canonical inverse correlation, anti-correlation between gene activity and promoter methylation. So the conclusion is, if you can target these genes, including some promoter regions, and find out about their SNPs and indels and structural variants, which they also talk about this paper, as you would do normally with Illumina sequencing. But on top of that, you can also find out about the, in this case, epigenetic mark of methylation in CpGs. Then you can use this to differentiate between tumorigenic and non-tumorigenic states. And here is an example of the remarkable change here. Methylated CpGs in red between this cell line and unmethylated CpGs in blue in this other cell line. And as you can see, in other parts here, they look quite similar. Maybe there's a bit more methylation here than here, this bar in blue. But in the promoter itself, marked difference between one cell line and another. And the other way around here, blue unmethylated and red methylated, right? So very visual result of what they get in these results. So how they did the sequencing and the analysis, what can we say? This starts to looking normal to people that are following the developments of nanopore technologies. Flow cell 9.4.1 using the grid ion sequencer. standard prepped. It has to be a PCR-free prepped, obviously. Otherwise, if you amplify your molecules, it erases the epigenetic marks. That's also the reason why you need enough material to put into the sequencer directly. And then in the analysis, they did base calling with Guppy that generates FASTQ files without the methylation mark. And they mapped it to with Minimap or NGM-LR for SB calling, but Minimap for everything else. And then they did the CpG methylation calling with nanopolish. And so kudos here to Winston Timp and Jared Simpson, who have worked very hard, and the rest of the people involved in having a piece of software that can call methylation directly from Nanopore data. This is a big feat of engineering, and the software is not trivial to write. So it allows the kind of science that is discussed in this paper. Conclusions, there can be improvement. I can see how this can improve quite quickly on the target rate, and this will open up more applications because it will make the per sample cost lower if the target rate goes up. 3,000 nanograms per sample, it compares to whole genome bisulfite in many ways, but it will narrow it down to certain applications. If this number of starting material can go down, and I know Clive Brown and Oxford Nanopore has shown ideas and internal R&D projects that the company has been working on to lower the amount of DNA required to load into the sequencer, then this will open up more applications. To put this in context, if you had 3 or 6 picograms of DNA, in theory, you should be able to get a full whole genome sequence at 1x of the human genome without any losses. So that's 3 picograms. 3,000 picograms, which is 3 nanograms, would give you enough, or 6 would give you enough for 1,000x coverage. So this is a 1,000,000x coverage. So without any losses, sequencing enough, they should be able to get 1,000,000x coverage on each region of the targeted regions, which means, they spend more money on bigger flow cells. Maybe they could have reached that, but with an on-target rate of between 1.5 and 7.5%, then you end up sequencing a lot of non-targeted regions as well. So the two go together in that if these numbers improve, it will open up to more applications. And then a final point, as someone who has been involved in epigenetics of other funky marks that are not 5MC, Nanopore themselves are also working on this training software with reference data sets for other modifications, not only 5MC. This will enhance the epigenetic signal obtained directly by Nanopore sequencing and analyzable, to get biological insight out of this sequencing technology. So this has been the presentation for this paper, this preprint, the analysis I made, and I welcome you to follow me if you still don't do on Twitter. And my Twitter handle is albertvilella, one word, twitter.com/albertvilella, one word. And you can also follow me on LinkedIn, ask questions, and I'll be happy to answer them. And as a reminder, this slide deck is available upon request. Thank you very much for listening.